Joseph Beechem has served as NanoString's Chief Science Officer and Senior Vice President of Research and Development since April 2012. Prior to joining NanoString, Dr. Beechem held various positions at Life Technologies, and most recently as Vice President, Head of Advanced Sequencing and Head of Global Sequencing Chemistry, Biochemistry, and Biophysics from January 2010 to April 2012. And from 2007 to 2012, he served as CTO of Life Technologies. During his career at Life Technologies, he led the design and development of multiple genetic analysis technologies, the latest advanced solid sequencing technology, and the single molecule nano DNA sequencing technology. Prior to joining Life Technologies, Dr. Beechem was Chief Scientific Officer at Invitrogen, and that acquired Applied Biosystems in 2008 to form Life Technologies. And so prior to his industry experience, Dr. Beechem led an NIH-funded research laboratory for 11 years. As a tenured associate professor at Vanderbilt University, he has authored and co-authored more than 100 peer-reviewed papers in diverse fields. And Dr. Beechem is also named on nearly 40 US patents or patent applications and has served on a number of editorial and scientific advisory boards. And he received his Bachelor's of Science in Chemistry and Biology from Northern Kentucky University and PhD in Biophysics from the Johns Hopkins University. And without further ado, we will turn to Dr. Beechem. Thank you.
Thanks, Ahmet. Good grief. When you get this old, it takes a while to get somebody introduced. So anyway, it's fun to be here. And I'll try to leave a bunch of space and, put questions in chat. It's hard when you're presenting in presenter mode to see them all, but Ahmet, just reach in and interrupt me if something comes up. And I want it to be real interactive. I'll try to get done enough time to have some Q&A. This is my labs up here in Seattle, Washington. You can see there on the left. We IPO'd in 2003. I was part of that IPO group. And my labs are there in that little building. We got about 200 scientists that work in my lab. We basically have two platforms. The one that we just launched, the Genomics Spatial Profiler up on the right. And we have an FDA cleared multiplex RNA single molecule counting technology. It's called Prosigna. That's actually, we just licensed those rights out to Veracyte. And then we have a normal multiplexed single molecule counting non-spatial result technology called nCounter. So we have these two platforms. Actually, here I am today. I hope the Wi-Fi holds out. This is a picture. I woke up this morning on a place called Lake Shasta, right underneath Mount Shasta in Northern California. So I was on a boat. So I opted into some kind of crazy little hotel room with Wi-Fi. So hopefully it doesn't go down on me. And then I'll go back onto the boat here in about right after this talk. I'm going to do a little something different than I normally do. I'm going to sort of show you what I'm going to show you at the end of the talk, hopefully motivate you guys to hang around for this thing. This gives you an idea. On the right, you see 1 square centimeter of tissue. Everywhere you see a little round circle, I'm doing spatial whole transcriptomes as I wander around that tissue. I forget on here, I got a couple hundred spatial whole transcriptomes done. It's a single FFPE slice, so it's a 5 micron thick biobanked FFPE slice. And I'm just going to wander through here and look at how the whole transcriptome changes. You got to wait till sort of the latter quarter of the talk, and I'm going to tell you all the science behind it. But hopefully this will motivate you to stick around through all the boring stuff like how we do it and all that. I actually started working. Well, it was great. I mean, Ahmet read. I sort of lived through the whole next-gen sequencing revolution, helped build these next-gen sequencers. And, we thought that we were going to solve every world problem by taking these tumors, blending them in a blender, to make a smoothie out of them. And then we were just going to sequence them to death. And we thought that we were going to get all the answers to the world's problems. It was great. We made TCGA and a bunch of the other databases. We moved biology in a real big science way forward. That sort of was the beginning of what I call big science programs. But the fact is that it didn't actually solve some of the fundamental questions, certainly in cancer. And that was due to the fact that in cancer, it's all about how the immune system interacts with the tumor, and the immune system's a game of rare cell stuff. So then in 2012, a technology called single-cell RNA-seq came along. It was a great technology. It got us out of this blender smoothie mode and now got us into the place where we could look at rare cells one at a time. And it was great. We saw a lot of things that we didn't see in the past. Now, the thing that's so frustrating, though, is you spend all this money on all this single cell data and you end up with a parts list. The thing is, you don't know how the parts fit together to actually make biological function. So that's why, well before a lot of the other people started thinking about the importance of spatial, I started working on a new technology for spatial. And I'll show you exactly when I really started thinking about it. It actually works out about 4.5 years ago. I started thinking new ways of doing spatial high-plex biology. And I'll show you, and that became this thing called geomics technology. We launched it in 2019. Actually, I was giving a talk. Actually, this whole work from home thing is crazy. You're not going to all these meetings. So I was giving this talk in Asia Pacific. I was sitting in a lab in Seattle, and I get this little chat question comes up, and it actually from some lab in India, and little chat thing comes up. Beechem, can you find me the little napkin you say where you invented this thing? I got better things to do than try to find that. But work from home, I actually went through my old photos. And what you see there in the middle of that photo dated January 7, 2016, I was waiting to give a talk at MD Anderson. So I'm in the hotel room of the Marriott that's right there at MD Anderson. And I remember scribbling down this idea for a new way to do high-plex spatial work. And that became the essence of this platform I'm going to talk to you today. But the essence is the same. It's an ugly little memo pad, but that's the way things work. You can see little antibodies I drew down there on a tissue slice. And those little squiggly lines are optical barcodes. These are not optical barcodes, these are sequencing barcodes, little 60 base pair pieces of DNA that I'm going to covalently link onto affinity reagents. And in this case, it's antibodies, but it works the same with mRNA fish probes. And then the trick that I was thinking about and what we ended up using is that we put those little barcodes on the affinity reagents with a photocleavable linker connector. You say, well, then what good does that do? You think about, what do microscopes do for a living? They shine light in very tight spaces. So I thought we could build a unlimited high-plex microscope where you don't put the assay in, you photocleave the barcodes out. So that became the heart of this thing called digital spatial profiling. And I'll show you exactly how we did that. Actually, so I scribbled that thing out on a memo pad and then worked it up later on that same day with a professor that was at OHSU. He was the guy I was doing all the protein barcoding with, a guy named Gordon Mills, who's now moved from MD Anderson up to OHSU. So I still do a bunch of protein work with Gordon. But that's how this thing originated. When I was interacting with Gordon, he said, he works at MD Anderson. He's got to make something relevant for human health and the human condition. So when I interacted with him, he said, Beechem, don't give me some technique that only works on fresh frozen because I can't do anything with it. It's got to work from the get-go, high-plex on FFPE biobank samples. So, and he was right. Everything I do was actually designed around making it work on these formalin-fixed paraffin-embedded samples, because that basically those FFPE samples hold the history of the entire globe on the medical history of the entire planet. It's actually about 12 billion of these slides float around the world. They're just sitting there waiting to be discovered. All that information just waiting to be tapped into. So anyway, we can do all the other samples, but I really did this with FFPE in mind. You can see on the left side of the slide, those are the affinity reagents. So we're talking about fish reagents, or you're talking about an antibody. The little barcode is with a photocleavable linker attached. They're about 60 base pairs. You can see my little napkin became the centerpiece of this particular marketing slide that's how it works. Now, the one thing to notice is that these reagents over here are invisible. You can't see them. So what we do is we dope in all these invisible reagents, and you'll see we're doing 22,000 at a time fish probes now. we'll dope in about four colored reagents, whether they're fish probes or colored antibodies or whatever. And on the right side, it allows the instrument to see the overall tissue geometry with its eyes, which is a camera, or your eyes, which are your eyes. And you can guide where the microscope goes before it shines the photocleavable light, and then you can actually see all the invisible, say, fish probes on this little region of the tissue or that little region. And I'll show you how we automated this whole thing, but that's the concept. So here's when you do these experiments, you got profiling reagents where all the invisible things are. You have imaging reagents where all the four colors are. They go in there at the same time. The imaging reagents guide your selection of what to look at. So this is the cyclic form of that on the right side. Step one, those two different colored reagents and the invisible ones. Step two, the camera can see these, your eyes can see these, and you can guide what areas you want to look at. And we do it in a very unusual manner. So I'll explain that in some detail. You find an interesting area, your photocleavage light goes on. It's actually violet light. People think it's ultraviolet, but it's violet. It's about 385 nanometer light for about a few hundred milliseconds, releases 100% of those photocleave barcodes, and it is 100%. And then the next step, 3 to 4, there's a little microcapillary that just literally drinks those barcodes right off the tissue surface. Your tissue's sitting in a little water bath, a little tiny liquid layer. That microcapillary just drinks them off of there, puts it onto an on-instrument microtiter plate. The instrument's on the left-hand side there. You open it up, and there's a microtiter plate in there. It drops these, what I call digital replicas, because those photocleave barcodes are the identity of all the affinity reagents. Your affinity reagents are still in there. They're in situ. You don't change the property of that tissue slice at all. It's all in situ, non-destructive. All I'm popping off are those little sequenceable barcode things. I call those digital replicas of the affinity reagents in that little microtiter plate. An entire operation there, whatever, 3, 4, 5, 6, takes about 30 seconds, 45 seconds. And then I just wander through the tissue slice, getting these digital replicas as I move through. And then when I'm done, I just pull the microtiter plate out, up to 800 plex. I read them on normal NanoString encounter technology, which can read up to 800 plex single molecule barcodes. And when I go over 800 plex, I got it all set up so you can just run them on an Illumina sequencer. And currently we go up to about 22,000 affinity reagents at a time now, and with an Illumina readout. And I'll show you some of that in detail. Any last minute questions here before I jump into one more sort of how-to thing?
So this is simultaneous to protein and RNA? Both
protein and RNA, that's right.
How about the quality of RNA? Because proteins require...
I'll go through that. It's really darn forgiving, and then I'll show you why in a bit. Yeah, so which means that, I've measured RNA on biobank samples that's 25 years old. Doesn't mean all of them. The key is that if you got a 25-year-old biobank tumor that hasn't been cut, those are ideal. Because you can take a 24-year-old tumor, cut the thing, put it on a slide. The clock really starts ticking when you cut them. And if you store them fine, the clock is really slow. But a lot of people just cut them and sit them out on a windowsill or something. But you can look at really old samples. Now, let me tell you, it's an unusual thing. This is something that's hard for next-gen sequencing people to get their head around. They're used to microscopes being dumb things. All they do is they raster something, or you're melting something in a matrix. And that's not the way this guy works. And I'm looking at chat things here, and I'll get my glasses on so I can read these things. How do you get a non-specific photocleaving? Okay. Okay, do you get any non-specific photocleaving? No, it's actually, there's a little window around your photocleavage. So if you look here in the back plane of the microscope that's geomics, there's two small semiconductor chips. These are called digital micromirror devices with about 1 million mirrors each. And actually, when I first built this thing, I had a single chip in there. And then the leakage of the UV light through the mirrors was just a little bit more than I wanted. So then between the alpha build of this thing and the beta build, I actually ganged 2 digital micromirror devices together. So the pattern is reflected twice and you get really pure violet light that gets put onto the sample. And the leakage has a full width half max, if you think of it that way, of on the order of about 1/10 of a cell. So it's really pretty pure. Now the interesting aspect is that I don't shine the light down in some kind of dumb manner, if you will. It's not like rastered. So this is the beauty of actually having a microscope that has a feedback loop between the profiler, which is the violet light, and the image, which is the 4-color fluorescent image you see. So this is how it works. So right in the center, you see a typical, it's a colorectal cancer sample. And one of the imaging reagents is a pan-cytokeratin antibody with Lexafor 594 on it. So you see that yellow signal is unique to the tumor cells, okay? And then the magenta purple thing you see is specific for the tumor microenvironment. So I take that picture, and then when I go profiling, I don't just try to average raster my way through this mixture of tumor and tumor microenvironment. I actually take that yellow picture. That yellow picture gets binarized, if you will, in the upper right. That becomes the mask that drives the semiconductor chip, which is going to distribute the violet light. So what you see is that, and I can do this in real time, so the mirror auto-configures to the exact tumor geometry that's in the tissue section itself. And then when I go shining the UV light, it only touches with single cell accuracy the tumor specific cells. And then I end up with a pure, say, whole transcriptome that's only for the small area where those tumor cells are at. Okay, and I put that in one well with a microtiter plate. So that's a whole transcriptome on just the tumor cells. And then the mirrors will invert. They do this automatically if I ask them to do that. They'll invert. And then they get a pure whole transcriptome on just the tumor microenvironment. So there's like 2 30-second operations on a small region, say 100 by 100 microns or so, of that tumor, that tissue sample, and then end up with pure transcriptomes of the tumor, and right next to the microtiter plate next to it would be pure whole transcriptomes of the tumor microenvironment, which is predominantly the immune cells. And those 2 operations took me about 30 seconds a piece, and then I can just waltz through the tissue section generating this kind of high-plex information. That's a long-winded version of how the thing works. And I'll try to continue to emphasize, okay, there we go. Those DMD chips configure themselves in all sorts of interesting ways. And when I built this instrument, I gave a hook out. There's a hook out, which means that you can do whatever kind of tricks you want in ImageJ with these 4-color fluorescent images, do all sorts of manipulations, and then you have control of the DMD yourself. So you can stuff in whatever mask you want to make. These can be crazy, complicated things. We programmed in a whole bunch of simple masks like segmentation, rare cell, and contouring, but you have control of the chips themselves. And I wanted to be able to do that for people because I can't think of every imaginative things you may want to do with that. Okay, this gives you an idea. This is a real sample now. I remember these groups came to me. That's the inflammatory bowel disease you see on the left-hand side. And the origin of IBD are these little mast cells that infiltrate these tissues. And this group was working like crazy trying to dissociate those little red mast cells, get them out of the little sample, put them in a tube and single cell RNA seq them. And they literally spent about 3 years trying to do that. And then they saw this technology. He says, okay, they just sent me this thing in the mail. We put the IBD up on the instrument. This was pre-release. I could see the little red mast cells. And then the mirrors will auto-configure. So then you can see the thing on the right. Those are auto-configured. Those are the little semiconductor chips oriented with every mast cell that was in that region of the section. They shine light on there, and then we're doing things like whole transcriptome on just the little mast cells in situ. I never tried to pull them out. Then we did that. We did whole transcriptome. We also did high-plex protein profiling on those little individual mast cells. And then while we're at it, we do all the other stuff too. The little green area was a separate little microtiter plate well that we put all that information in. But it gives you an idea how this stuff works. This is your brain on DSP. So these are brain slices going in. And what you can see is that, see that little astrocyte on the right? The mirrors will auto-configure around a shape that's like every astrocyte's different. It'll auto-configure. And then what you see in the middle of the screen, I'm measuring 44 proteins on that single astrocyte. And I do it a couple of times because it's non-destructive. I can do that. And that's the reproducibility on a 44-plex on a single astrocyte. We typically do this now with about 100 proteins at a time. But that gives you an idea of sort of some of the protein work. And that's the most mature part of the work. I probably don't have to tell this group that. This is a spatial omics group. So what I have here is left and right screen. That little TIS thing, that was actually a clinical grade signature, RNA signature we developed with a company called Merck. And it was a signature to guide immunotherapy with a drug called Keytruda. So the higher number, and that's a bulk blender assay kind of thing that we did for them. So if I scrape that tumor on the left, I get a little bulk number, 8.32, and that would say that tumor could respond to Keytruda. And then you can see the one on the right, scrape that thing off and measure it. You get a similar number. Actually, my screen is, I got, I forget what number I'll move. Yeah, 9.13 there. They both would have been a bulk version that said they could respond to Keytruda. When you actually look at them spatially, they're totally different classes of tumor here. One of them is going to respond and one of them's not. The one on the left is not going to respond to Keytruda because there, the immune system is partitioned away from the tumor cells themselves. You can hit that thing with a checkpoint inhibitor, nothing really is going to happen. So, but if I had the bulk assay, I wouldn't have been able to tell them that. But the one on the right, you can see all the infiltrating immune cells. That guy's right. That thing's ready to respond to Keytruda, but I wouldn't know that unless I went into the microscope. So that's an example of why all of us are working in spatial biology now, because you can't average these things out and try to make sense of them. This is some of I'm going to go real quick here. This is some work we did with the pathology group at Yale. This gives you another example of the mirrors auto-configuring around a real cancer, in this case melanoma. You can see the signal on the left, the mirror configuration, that's just going to be the melanocytes. That's the tumor specific. And you can see how torturous these cells are in there. And then on the right-hand side, the mirrors are just tracking all the macrophages in that particular sample. And we did a couple of other compartments at the same time. The main thing we discovered with that, everybody talks about this marker called PD-L1 as being a good predictor. What we found out of that study is that PD-L1 is a good predictor, but its main predictive power is when it's on the back of a CD68 macrophage. But normally people aren't decomposing exactly where these signals come from. But here I had mirrors that were auto-configuring around the macrophage compartment. So we could test that. And it's actually PD-L1 on the back of those macrophages that was the most predictive marker. And that thing got us on the cover of Clinical Cancer Research almost a year ago today, actually. And I think that's sort of all these. I'm sort of going to stop on the protein side of here. This gives you an idea. To give you an idea, actually, when I first built these instruments, I made them under remote control so that we started taking samples about 3.5 years ago or so that people would send us in the mail, because FFPE, you just drop it in an envelope and send it. Really, it's so easy. And then I made these microscopes remote controllable so these scientists all around the world could actually literally move the microscope around, select their regions. And now we got over 25 papers that came out of these little homemade. I literally made these things. It's a typical academic research style microscope that ran. We now got about 25 papers. Our very first 2 papers were back to back in Nature Medicine. The author on our very first paper on this homemade DSP was actually a guy named Jim Allison who won the Nobel Prize last year for immuno-oncology. He discovered a molecule called CTLA4, which is actually one of the key checkpoints. It was fun to have Jim on our very first geomics paper. He was literally sitting in Houston, Texas, him and Pam Sharma controlling the instrument in my labs in Seattle. If you want to see the down and dirty on everything about the microscope, I think maybe this is maybe the one that caught Ahmet's eye. I know we had a recent paper that had all the data in Nature Biotech. So if you want to see all the crazy validation stuff that you got to do, take a look at that paper. It's a good one. And then everything else in between. Actually, we just had back-to-back papers in Nature the other day, which is kind of neat. But anyway, it's pretty mature. It's not this technology works. Now, when I built this thing, I really wanted it. It was built from the beginning, not only for FFPE, but it was built to be able to do RNA profiling and protein profiling. We believe that it's very important to have both kinds in there. And the microscope can allow you to do independent profiling on one serial section, get the highest sensitivity protein. You can have another serial section coming right behind that, high-plex RNA. There's software that'll align those two slices together, and then you can do all sorts of crazy, what's my protein signals doing, what's my RNA signals doing. You can actually combine them on the same slide. You take about a 20% sensitivity hit. If you don't mind that, you can do it on the same. It doesn't really matter to me sometimes. I actually like them on, I like the highest sensitivity assay term, but it's not necessary. Now, let's give you an idea. When you get into RNA, multiplex is unlimited. 2 years ago, we were the highest plex RNA spatial thing, and that was at 84 plex. That was about 5 or 10 times more than the leading commercial competitor at that time. And in the last 2 years, we've gone from 84 plex to 18,000 plex. We did the human transcriptome, and that launches early next year. And then we had a lot of people that were clamoring, sending me emails saying, we need mouse too. So I went ahead and did the mouse whole transcriptome. It's also going to get launched early next year. And that's a 22,000 mRNAs we look at in the mouse system, about 18,000 we do in the human system. They'll both go out the door early next year. Right now, we just launched one that's about 1,850. It's all for the cancer. What you find is for cancer, we know so much about the RNAs in cancer. We took all TCGA, dumped it in, all the most important ones. It turns out you need about 2,000 genes in space to do a really good job on everything that's known out of TCGA. And that's called the Cancer Transcriptome Atlas. That's commercial now. I'll show you some of that data. This is how the barcodes work. When you're going to RNA barcodes, you can see our little fish probes there. And when we were doing that, I set it all up so that you can run them on any of the Illumina sequencers, all the way down to MiSeq, NextSeq 550s, NextSeq 2K, HiSeq, and NovaSeq. So we designed these things with the I5, I7 double indexing that probably many of you have used. You see they got universal molecular indexing on the inside. There's 14 base pairs of UMI. So when you're in spatial biology, it really helps to have that really high horsepower UMI, because you can have huge dynamic range switches between the different analytes you're measuring. And then we got a 12 base pair indexer. And when you're at Hamming distance 3 and a 12 base pair indexer, you can go up to about 1 million different RNA molecules. And you can see right now we're just up to about 22,000. But that's how it works. Simplest kind of library prep you can ever imagine. All the little photocleave barcodes are exactly the same size. They all got the same indexing. They all have the same primers. So it's a single, simple one-step library prep. It's the world's easiest one. No biasing in it. They're the same. We designed it that way. Every area of illumination becomes one individual well in there. And then I typically run it through in my labs for like your own personal little geomics. I really like the NextSeq 2K right behind there with 1 billion reads. Works out really well. Here's the little indexing things. I put them in there for completeness. So it's just so later on you want to stare at exactly what we got going on there, and you can see exactly how many base pairs we have for these things. And we'll send you the sequences if you want to know what they are. And then the key thing is that the data comes out of the Illumina sequencers has to be remarried with the high resolution images. So that's really what we do. So the data photocleave barcodes go out, you sequence them, you know, Illumina had no idea that these are all spatial barcodes, right? So they come back as a whole bunch of reads. We wrote the pipeline that actually converts those Illumina reads into high quality digital counts associated with all the different mRNAs you're measuring. And then it marries it to the exact illumination volume and high resolution multicolor pictures of exactly that region in space where all those sequencing barcodes came from. So that's how we marry those two things. Here's this kind of sequence you can see. If you're doing proteins, you go all the way down and get, you know, your MiSeq can actually work before you if you want up to 100 targets or so. As you get into a couple of thousand genes, you can run it on NextSeq 550. As you depending on how many things you want to multiplex together. And then as you move, you can see when you get into the whole transcriptome, you're getting up into between NextSeq 2K and NovaSeq land, depending on how many things you want to do. But you know, you can see NextSeq 2K fits there. It covers almost the dynamic range of all you want to do. You know, if you're running tons of experiments like I do in my lab, then you know, we keep the NovaSeq busy pretty much all the time. But anyway, that gives you an idea of kind of sequencers. And now it's kind of fun. You know, we were in totally new land. When I started telling my own R&D scientists we were going to do 20,000 RNAs in space, you know, I run R&D, but they still look up at me and say, you know, you're crazy, Beechem. We can't really do that. And how are we going to measure that? Yeah, there was no book. You can't look it up in a book how you're going to validate that kind of stuff. This is a nifty one we did. It was crazy how well it worked. So let's give you an idea of what we did. You know, we got great RNA-seq libraries all over the place, right? And one of the ones I really like is the Cancer Cell Line Encyclopedia. That's put together by the Broad. And I think there's the little Nature paper on it when they updated it about a year ago or so. So what we did is we went in and made FFPE cell pellets, fake tissue, if you will, of all these cell lines. And then we're going to do a whole transcriptome or cancer transcriptome on them. And then I'm going to look at how they correlate with the cell encyclopedia library. And I'm going to do it in a special way. I'm going to do it blind. I'm not going to tell my people which cell pellets are what. So we put whole transcriptome or cancer transcriptome. And I said, just go against that database and tell me which one it was. There's about 1,020 cell lines in there. I gave them 11. And I said, you know, just nothing fancy. Just plot me out the highest correlated cell line. So you can see over on the left, my cell pellets have about 80 to 350 cells in them. They're going to get compared to RNA-seq data from these cells, same exact cell lines. It's about 300,000 cells worth. And then we're just going to project them on each other and say which ones correlate. And what you see there, and they got two different kinds of cancer lines. These are, you can see the blood-based tumors are all the little light blue ones. The solid tumors are all the little green spots. And you can see blinded, they caught every single cell line from 80 cells worth of whole transcriptome or cancer transcriptome. I could tell you blinded exactly which ones they were. So what that tells me is that the biology that we're following in these high-plex RNA things really follow the biology of a high-resolution RNA-seq, and it's covering all sorts of different biology types, whether it's liquid tumors, solid tumors, it doesn't matter. I was pretty happy with that kind of correlation. And then what I had them do, I said, okay, do the entire thing twice. I want two different users doing it, running two different things, running two independent library preps from scratch. They independently sequence, and then we compare the numbers with each other. And I knew that the digital barcoding is incredibly accurate, but I didn't know what kind of noise was going to come out of the next-gen sequencing library side and the next-gen sequencing readout. And this is what you see. Those are all the cell lines done, two users, totally independent libraries. You plot the raw counts on top of each other, and that's it. Greater than 98, 99% reproducibility in here. This was nuts. I remember I was at AGBT showing, I was showing Gary Schroth this. I don't know, some of you may know Gary. He's sort of like Mr. Sequencing from day zero at Illumina. He saw this thing and he just about jaw dropped. He says, that's independent libraries, independent, the whole process. I said, yeah, Gary, that's the way it works when you're counting barcodes on these things. These are all FFPE slices. So we actually, we had a lot of fun together. We tag teamed up. We've given a bunch of NextSeq 2K combo geomics talks and a whole bunch of things. It's been a blast. And you can YouTube it if you want, just sort of search Gary Schroth and Beechem, and you'll find some of our talks online if you really want to see the down and dirty on how these things work with the sequencers. Okay, that's sort of the boring how's it work kind of stuff. Let me show you what you really roll up your sleeves and let's have some fun with some tissue slices. I'm going to show you what a couple of thousand specially designed cancer transcriptome atlas kind of things. This gives you an idea. This is shipping. Nice thing is with TCGA, we have so much information. I can categorize all the things that are happening. If you actually look at TCGA, what's going on, you got immune response. We got a whole bunch of genes. We're going to link to that. You got tumor biology and you got tumor microenvironment. That's sort of the high level categorization of what we did in CTA. Then you go one layer deeper. In there, you look at signaling pathways, cell function, immune response, innate, adaptive, metabolic responses. That's in there. You want to go down farther at the pathway level. You can see we pow in whatever it is, about 115 pathways linked to all of these things. We do high level tiling, so you get super high sensitivity of the measurements that you're doing in space. This is sometimes people don't realize this. You know, if you're doing this kind of high-plex RNA and all you're doing is like poly-A pull-down stuff, you get killed. You get killed by the dominant RNAs, right? You can't see the stuff you're really after seeing. So when we design this transcript, there's no poly-A pull-down. These are hybridization things that go right in situ to whatever area of the gene you want. So when you do that, you don't get killed with all your sequencing money going down the drain, sequencing actin and GUSP and all these things. You go right at the key immuno-oncology and pathway targets. So when you actually look at the CTA that we're doing in space, and then you go against something like TCGA or whatever kind of cancer transcriptome, whatever kind of cancer database you want, you can see that we can see high expressors, we see mid-level expressors, we see the low expressors at high efficiency. And these things aren't invisible. So you really do reach in there and grab those. If I'm doing poly-A pull-down stuff on these tissues, about 80, 90% of my signal are up there in all those high expressors. And most of them aren't even here. And I'll show you how we got rid of them. We took those things out. We took them out of our whole transcriptome too. So if you want to see deep, this really helps. That poly-A thing really kills you when you want to look deep. This is that seminal publication. You want to see the down and dirty. This is what you want to look at. It's kind of fun. I put a little bit of the protein stuff in there. It's kind of fun. These little mirrors allow you to like shoot bullseyes, and I can just digitally quantitate like what happens if I shoot the mirror at one cell, what happens when I shoot the mirror at 2 cells. That's that little linear graph, 1 cell, the second one from the top. That's 1 cell done about 4 or 5 times. 2 cells, you move up on the linear axis, 4 cells at a time, 8 cells at a time. The dynamic range is nuts. You can do about 10 to the 6th with these mirrors going up, digitally counting these barcodes. Your assay, crazy linear. And then on the right is some of the high-plex RNA I was doing, back then. And, you got multiple thousands of RNAs. I don't even know what half of them are. I know they are important. They come out of the cancer database. But what you find is that I remember when I started showing this raw data, we were just doing it for validation work. What we found is that we had all sorts of discoveries in there. And what we found is about 10% of the discoveries are already validated in the literature, right? And this is one of them. I was showing this data at an AACR meeting, and one of the leading immuno-oncologists, a guy named Jerome Gallone, he came up to me, he said, Beechem, I'm looking at the data. there's an anomalous finding that your microscope, your geomics finds just like my studies did. Turns out there's a little chemokine called CXCL14 that has a weird backward behavior and with respect to the immune response. He said, Beechem, that thing worried me so much. You know, I was anal. I did qPCR to replicate that, RNA-seq in detail to make sure that was a real result. We published it back in whatever, 2016, made the cover of Immunity. I didn't even know what CXCL14 was. And he said, look at your geomics data, it's exactly that result that we found in 2016. That gave me additional confidence that what we're measuring here is real biology. And we've continued ever since this day now for about the last year and a half. Every time we measure, we discover stuff. About 10% in the cancer community, you can find an original lit version of it, but the other stuff's just brand new. It's all there for the taking, actually. But anyway, now let's go into something a little deeper. This is really fun, and I apologize for all the cancer immunology because that's sort of what we specialize in. You can play the same game in brain with Alzheimer's disease or watching little mice develop in the microscope or all those things, but bear with me. This is the area I really know. Let's take a look at this. This is it's a colorectal excisional biopsy, and that's, you see that in green. Those are all the tumor cells. You can see the immune cells there in magenta and white or whatever, and it's a war going on here. Okay, and your life either depends on who wins the war. The immune system wins, you get to live another day, and most of the time the tumor wins here. So you really want to understand the spatial biology of this crazy war that's going on. And let me show you how you use these specialized mirrors to really help you with that. So we're going to do cancer transcriptome profiling on this war, and we do it in a very special way. We focus in on where the battleground is. This is right there, right at the interface where the immune cells are sort of attacking, if you will, the tumor cells. And I'm going to configure the mirrors in a really unusual way. I want the mirrors to follow the geometry of the battleground. This is not little individual cell stuff. This is work that's happening along a boundary. So what we do is we auto-configure the mirrors, and look at how we auto-configure the mirrors. Those are 50 micron line, little contour regions. And we are going to follow the war in the spatial axis as it happens. It happens along this kind of geometry. Every tissue is different, but the boundaries are all there. You always get these two things conflicting with each other. So every boundary is different. That's why we got a semiconductor chip that has infinite degrees of freedom. And then I'm going to probe the biology at all the pathway level along that spatial contour boundary. So let's see what it looks like. Okay, there's the little boundary. You can see how the mirrors have set this up. I'm going to do a couple of things. I'm just going to look at the data. That MET gene is a gene that should only be found in tumors. So what I should be able to see as I look along that gradient, it should follow the tumors. And then the minute I get off the tumor cells, the expression level should go down, and that's where you see the whole thing turning blue. And we're going down into a deep ocean. There is no MET out there, and then it just goes blue. Okay, so that tells me, okay, I can follow that thing along a spatial gradient. Now let's talk about some interesting one. My favorite one is look at MHC class 2. And this is really cool because I think this is really how the fight plays out. Remember, right to left, you got the immune cells coming in, and right where the little hash line is where the immune boundary and the tumor boundary hit each other. Those little contour regions are 50 microns. So as the immune cells move from right to left, they're coming in on that boundary. You're getting close to those tumor cells. And when they get close to the tumor cells, you're going to get all this antigen presentation stuff to go on. So that's how the immune cells work. And they're going to start presenting class 2 antigens on their surface. So they got to ramp up the mRNA. So as those immune cells come in and hit the tumor, you can see that burst of pathway activity right in that key area where these two groups are fighting it out. And they're fighting it out. This is how the war gets played out. The immune cells start showing all these tumor antigens up, start to get activated, and then you get all the action on the other side of the fence there as it starts working its way in. So I think, this is the literally 4.5 years ago when I was down at MD Anderson sketching out where we wanted to be. This is what we're sketching out on that whiteboard. It's kind of rewarding after whatever 3 years or 3.5 years. You can watch these battles take place just like this. It's really cool to see. I tell people we're gonna, and this is stuff that's just not known, all the stuff, how all the pathways go, but this is what you can get every day now. And I tell people, it's time to rewrite the textbooks on how these systems play out in space, because we've never done it this way. Either people have just averaged the whole transcriptome over these kind of games or these boundary areas, and they all get averaged together. Or regrettably, we got some technology. They're great technologies. I don't mind them, but they don't follow the geometry of what's happening. The tissue architecture does not follow an XY coordinate frame. So if I'm making little arrays and put little spots on an XY coordinate frame, it's fine, but it's not following the geometry of what these tissues are doing. And the signal and noise really matters on how you follow the geometry. I can amplify all sorts of low messages that are just hugging the boundaries. And if I just put a single spot or a single pixel on that, I'll never see that thing, because some of these are rare messages happening. Okay, I'm going to end this thing in about 3 minutes. Let me show you a little bit. When you play the game, whole transcriptome is the same game. It's over 18,000. It's about 18,500 genes. If you find something that's not in there, and you can custom put your old barcode in if you want. Now, when we're doing this, if anybody's ever done this very much, you realize you waste a whole lot of money on high abundance genes. You got mitochondrial genes, you waste all that stuff. It really isn't helping you a lot. If you want them, you can custom make them. We took that stuff out. When you take those the top 10 high abundance genes out, some of the mitochondrial things, what you do is you save about 33%. A third of your sequencing cost just disappears. Who wants to spend money on GUS-B? I didn't want to. We just took that stuff out. So then your whole transcriptome is really on stuff that matters. It's like 18,500 genes that really are going to carry some weight in it. It's not poly A pull down, so you're not limited by the abundances of those things. But that's how we set it up. We ran whole transcriptome through the same blind study we did for Cancer Transcriptome Atlas, correlated with all the cell cycle encyclopedia thing, and it works fine, as I expect. Now, just re-emphasizing, we're looking at whole transcriptomes now. These aren't averaged whole transcriptomes. They're transcriptomes in key niches. Tumor microenvironment typically, but if I'm in the brain, it may be the whole transcriptome of an Alzheimer's plaque, and then the thing is the 50 micron region right around the plaque, say, as an example of something you do in the brain. Okay, key thing is we didn't know when you're doing FFPE, this is a colorectal excisional biopsy, I think it's about 5 years old. How many genes of the whole transcriptome are you going to see? And this is what we measured, and it turns out that we average about 9,000 genes that you typically measure on any particular region of say a colorectal cancer. That's sort of an area I wanted to be up in that region, so that was good. Now these are segmented 9,000 genes, so you get a lot of extra information out of those. They're not just averaged. This gives you an idea. You can play a game like this. This is actually one of the first ones I remember doing. 1 centimeter by 1 centimeter. I got about 300 compartmentalized, so 150 transcriptomes of the tumor and about 150 transcriptomes of the tumor microenvironment partitioned along 1 square centimeter of tissue like this. It's about 5 billion reads, so you soak up a NovaSeq run if you want to do it. But this sort of gives you an idea. If you're really interested in some deep information on a 1 square centimeter, you can have at it. To give you an idea how much time it takes to do an experiment like this, you do the whole thing on the geomics will be done in about a day and a half. So you can go pretty quickly and get this kind of information if you want it. Now, what do you get when you do this? Here's that same colorectal. And then we made some visuals that allow you to just literally watch your transcriptomes as you move around. So I made these circus plots, okay? And I'll give you the detailed anatomy later. All around there, we organize the pathways in a circus kind of thing, circos, or however you say that. But again, you'll get these pure transcriptomes. You can see on that circos plot, I'm going to have the tumor stromal area is like yellow or orange, if you will, and then the tumor-specific transcriptomes come up as little green spikes. And then what I'm going to do is I'm going to wander through the tissue, watching how that changes. And you get some people saying, well, I call it the deer in headlights. You do this kind of high-plex. How are you ever going to make sense out of it? And that's bogus. I never believed in that. You got high-plex information. It's just how you organize it. And this is so easy when you organize it this way. You can actually do the analysis yourself from the back of the room. So you can see how at partition inside the circos plots, you can see I can organize the pathways around the clock. So like whatever it is, between 12 and 1, you got a whole bunch of cytokine stuff, and you just organize it that way. Each little spike would then be the raw next-gen sequencing coming out of, say, a particular gene in the cytokine pathway or whatever. We're just grouping them together. So you can actually look along, and then you end up with 2 compartments, the tumor microenvironment. And then let's just literally watch a couple of these circus plots, and I'll help you out a little bit. So let's just take a look. You see those 2 little regions that we looked at there? They pretty much look the same. From a pathological standpoint, sort of a mixture of tumor microenvironment. They're not that different. But then look at the little circles plots for, you're looking about, you're about 1/4 to 1/8 of a centimeter away from each other, those 2 little round circles here. But then you can look at the cytokines on the one to the right, a flaming hot cytokine bunch of stuff going on there. I'm moving 1/8 of a centimeter to the left there, and pretty much the cytokine stuff's down, nil almost. And that just gives you an idea on how these things change. Let's do another one. Let's take a look at over there. I see some stuff coming up in the chat. Let's see if I can take a peek at some of these things. Okay, I'm going to continue, but I got my little chat box there. I'll be able to look at that. You look at the right-hand side. Look at that little guy, circus plot there in the middle on the lower right. Yeah, cytokine's pretty hot. But then look at that there. You're down in the cell cycle and cell-cell interaction part. That's all down. I don't know. This is just the way the biology of this tumor is going, this tumor slice. And then you can compare that to the region that's just whatever, a spot, 1/4 of a centimeter away there, maybe 1/2 a centimeter away on the other side. What I'm trying to do is give you the flavor that you talk about doing a whole transcriptome on a slice, and you got to ask yourself, what transcript? It's an average of all this stuff. And here you can see that these things change on like all you can go 1/10 of a centimeter away, and whole pathways are going up and down and changing. Okay, let me read a little bit of this chat. We're getting pretty close to the end here. Okay, let's see what we got here. Okay.
You can answer after you finish your talk, I think. Yeah, so
okay.
People can just put a question in the chat box now, but I think Dr. Beechem can answer in the end.
Yeah, that's great. That's great. And we should try to maybe Ahmet and Ron, maybe can you save that? You can save the chat file and send it to me, and I'll make sure and get back to everybody if we don't get to everything. Okay, we're pretty close to the end, so I think we can open it up for a dialogue here. But anyway, this gives you a feel for it, okay? Now, okay, let me get back onto this screen so I can advance. There we go. Obviously, people don't do analysis by eye, but, it's reassuring. These signals are big. You're not like fighting to see these changes. I can see them with my eyes, which means when I go in and do my algorithms, the real math on them, they shine up like huge stars, right? This gives you an idea of volcano plots of all the pathways in the human cell. So every little dot is an individual pathway. So you can actually see which ones are changing between any 2 areas. And then you can do things like map out which pathways are, 1/10 of a centimeter this way versus 1/10 of a centimeter that way. And I'm sure there's a lot of people writing fancy stuff for this. So I look forward to using that stuff. And now I'll tell you one more story and I'll be done. that's a story of a army. It's a battle happened on a battleground of spatial dimensions of boundaries. There's another cool story that happens on the individual foot soldiers. As those little T cells migrate on the inside, I wanted to follow what the heck happens to those individual foot soldiers, if you will. So this is a case where I configure the mirrors around the individual T cells as they go from that stromal area penetrate the tumor. I wanted to follow them in. And then I wanted to build little neighboring boundaries, about 10 microns, 15 microns around them, and say, okay, what happens to those troops as they go in? They're surrounded by the enemy. What else going on? So we independently measured that cell compartment and then this nearest neighbor compartment. And you see some pretty cool things. I'm going to focus mainly on the right-hand side. Okay, let me take you through the CD3 signal. You know, when I'm way out in the stroma, you know, I should see pure T cell stuff. When I do my little neighborhood stuff, I shouldn't see much other T cell stuff because, you know, it's pretty clean segments. And then let's look at the middle one, the TIM3 one and the 41BB one. So what you're looking at is in the stromal area, maybe I think TIM3 is kind of a neat example. When you're way out there in the stromal area, it really doesn't matter. That nearest neighborhood of the T cell is pretty innocuous. There's not a whole, that T cell is not tired yet. It's not worn out yet. It's not exhausted yet. But then when you follow it in, so when they go into the intratumor area, what you see is you see some exhaustion markers just pop up. TIM3 is an example of one. 41BB is an example one. These are soldiers that have been fighting a heavy fight on the inside. Whole bunch of extra markers come up, basically as like signs of exhaustion. It's a good way of actually seeing some of these soldiers wear out as they actually try to complete their mission. This is where the checkpoints come in, and all the drugs that are now combo drugs related to TIM3 and a whole bunch of things. But anyway, it's not an IO lecture, but it gives you an idea how you can follow these things. And I really want to end. We're just getting close to the thing. I won't go through this story. I'll just leave it in there. When you have these high-plex things you can do in space, I call it solving the lost in space problem. If you're a single-cell RNA seeker, you end up with all these great UMAPs and you don't know where all the clusters go. When you got high-plex geomics on the other side, you just go ahead and make your high-plex RNA measurement, and then you basically can deconvolve those signals off of your UMAP, and then you can solve sort of what I call the lost in space problem. All of a sudden, I can start finding where your little single-cell clusters from your dissociated tissue experiments go when you actually look at the real tissue. This is work, we've done this with a couple of investigators now, Alex Swarbrick from the Garvin it's in Australia. He's actually one of the first people to get some of the whole transcriptome stuff, and he deconvolved a whole bunch of interesting things out of there. If you're interested in the lost in space problem, send me a note. This sort of summarizes it all. I think you guys got the message pretty well. I won't spend a ton of time here. You know, you can do a lot. You can detect a lot of protein targets. We got about 66,000 antibodies you can play around with. A whole bunch of them validated. I think inside our validation shop is hundreds of them. And then Abcam's got 6,000 antibodies that you can barcode. You can look at any kind of IEs, if fixed paraffin embedded. You get these weird regions that you can look at where your microscope is active. And then you could do protein or RNA. And you can see it's a reasonably mature. It's not mature like PCR or a sequencing box. So, you know, these things aren't, you know, we still, you know, it's leading edge kind of stuff. I think it's a good place to end. We can open it up. Thanks for your attention. You got some crazy ideas on stuff you want to play around in spatial biology? I'm always looking for them. And let's take some questions.
Thank you, Dr. Beechem, for this terrific and great talk. And now for questions, either please post it on the chat or unmute yourself to make it more interactive. I think there's one, it's not a question, but it's a note from Dave Nguyen. So he says, my algorithm, the numerized histogram score, can quantify bar plot distributions in the form of one number. So
Yeah,
basically there's an
I like it. Okay, I'll definitely look at that. We're always looking for scalar indicators of some of these effects. Because, when you're interacting with, you got some pathologist on the other side of the equation here. In the end, we know how to do, I'm saying we, I think most of the people on this call probably more technology style people, but in the end, you got to tell your results to some pathologist or some clinician somewhere. Having a scalar number like that's really helpful. So I'll definitely look at that. Thanks.
So hi, Joe. I think I know Gary very well. I remember last time we were on a beautiful island, and he was talking about how to kind of optimize the FFPE tissue processing to extract RNA. I think I hope you didn't upset him. I think because that means he just doesn't have to do that anymore.
That's true. We just it's a good point, Ron. Because it's worth emphasizing the RNA never comes out of these slices. Okay, it just lives in there. Yeah. It's in situ. The photocleave barcodes come back out. So actually, when you're done with this whole experiment, you actually can pull your little slide back out of the geomics. I stored the thing wet, and you can do it all over again. You don't lose anything. It's non-destructive. For proteins, I can pull that guy in and out like literally 5 and 10 times. RNA is not quite as good because the different RNase treatments and things you do for in situ hybridization do weaken the tissue. What we find is you can go in and out twice. You get exactly the same digital counts again. So you can do you could do 2 whole transcriptomes twice, or you do different areas and the same numbers. On the 3rd time, it goes down about 20%. So you could do another one if you wanted. After that, it does start going down a bit. But the nice thing about never pulling the RNA out is you can go back in and see where a whole new batch of it is. It's kind of fun.
Yeah, that's cool. That was actually the question I was about to ask. Seems like you already answered. You can actually run the same experiment twice, exactly the same tissue in the same tissue region. Since that's highly reproducible, maybe you can even use that to double the amount of targets you want to look at. Probably not necessarily for transcriptome, but for proteins, right?
Yep. What one thing I like to do sometimes, you'll get some statistician just wants to see it done twice. You find you give it to them. But I like sometimes you'll do a whole transcriptome on some region and you've cut it up in some kind of spatial boundary thing. Like let's say we're looking at that boundary game I was playing. Then you may say, you know what, I'm going back into that war zone and I'm gonna cut the thing up differently. I'm gonna maybe instead of contours like that, I look at it with strips like this, say. And then you could go back in there and do whole transcriptomes along strips and see if there's other things that you see. It's you can get imaginative here. But it's I just tell you, it's no technology is like a be all end all, right? I don't pretend that this guy is one either. What you don't get out of this technology is that it's not designed to do, single cells one at a time, 10,000 single cells all in their own little thing. That's not what this guy is designed to do. It's really about the biology of, big structural biology of these things. I think we'll have other tools that are come up, some of the, some of the other single cell imaging things that do a good job. They're much lower throughput. This guy will go through 10 or 20 whole FFP slides in a day. So you tend, when you interact with the outside world, and they have a patient cohort of 50 or 100 slides, it's not like, we can take that on and give you all the data back in a couple of weeks. If you're doing all those single cell things at a time, sometimes you get a slide done. If you get a slide done in a couple of weeks, you feel pretty lucky. But anyway, that's not a be all end all. It's really, you can see what the tool's been designed to do.
Great. I have actually two questions. One is technical and one is more biological. The technical one is, the subcellular resolution, like can you get down to, you know, some part of the cell specifically? What is the limit of your UV light focusing capability? And the biological question will be, have you ever tried looking at the immune checkpoints after therapies, before and after?
Yep. Yeah, actually, most of those papers are like before and after papers. So yeah, beautiful. That's really, yeah, I think our first two back-to-back Nature ones were that way. And before and after, another one was cool because it was a surprise. We were actually interacting with these T-cell biology experts at MD Anderson, and it was led by the pathologist and a lady named Jen Wargo. And yeah, she was doing some of our panels, our protein panels are about 100 at a pop, and had a whole bunch of these B-cell stuff in there. And she said, you know, I don't know why you wasted my time with the B-cell stuff in there. And it turned out she discovered the importance of what's called tertiary lymphoid structures, and it's the B-cell activity in these things called TLSs that she found that way. And now she's had back-to-back papers. Some of the ones in Nature were associated with that effect. So those were all before and after profiling like that. Now, subcellular resolution on these mirrors, the mirrors themselves are 1 square micron. So you can go down there. But I'll be honest with you, you're not going to see that with this technology, you're not going to see messages at 1 square micron. Now you can do things like you can subcompartmentalize and say light up all the nuclei. You could be profiling nuclei if you want. You could profile cytoplasm if you want. We don't do a ton of that, but you could do those compartments. But it's not subcellular at anything other than that. Yeah.
And then I think also Han Sao was going to ask a question. I saw them showing themselves. Is there any questions? Feel free to unmute yourself, guys.
Yeah, feel free to unmute or even kind of show your face. Yeah.
Yeah, absolutely. So, it's great to be able to. I took my screen away so I can actually see people.
Yeah, there's a fan club. I hope we have this kind of family-like feeling here. So if you want to unmute yourself and show your face and talk to Joe, and yeah, you're welcome to do.
Maybe another minute for questions.
So I think you mentioned in one slide, kind of you detect messenger RNA protein and a PTM. So
yeah.
what are you thinking about for PTM?
Yeah, that's post-translational modifications. Yeah. So I was really happy with that, actually. When we're doing proteins, and all these people tell me you can't monitor the phosphorylation status. But, you know, it's always good. You know, you don't always believe what they're telling you. So we went ahead and we had a good collaboration with Cell Signaling. Cell Signaling has some really beautiful phospho-specific antibodies. And then, so we put some of those in and said, could we quantitate the phosphorylation status of some of these targets? And we found that we absolutely could do that unequivocally. You find that it matters a little bit. When breast cancer is a great example. Breast cancer tumors are pulled out using either excisional biopsies, or there's another thing where it's core needle biopsy. What we found is that you go in with a core needle biopsy, you pull that guy out, you fix it, and make an FFPE slide out of that core needle biopsy, absolutely preserves the phosphorylation status of those markers in there. And it mainly does that because you don't have some surgeon in there tying off all the blood vessels and things, and before they get it out, they get compromised due to that. So we got beautiful signals on phosphorylation, and you can really, and we found some really neat things where from the genomic side, we were monitoring mutations from the bulk on like things like EGFR and all these things. And then we went into the tumors and we found classes of EGFR receptor modifications where they were predicting would affect the signaling cascade. And about 3/4 of them do, and 1/4 of them does nothing. So if you would have gone in there with some anti-phosphatyrene, some RTI inhibitor for that particular patient that didn't have the downstream signaling, you're wasting your time. And so you can see interesting things like that. I'm a believer in the PTM. We continue to work it.
This is amazing. This is truly amazing. I think people should not do Western blot anymore. Just yeah, you can do spatial protein
Spatial
phosphorylation.
Beechem,
Yeah,
absolutely, really cool. And yeah, we got some other new spatial games we're cooking in the lab, and maybe you'll invite me back again. I'll tell you the next generation of tricks. Actually, if you keep your eyes open, even where are we at in the month of September? Yeah, keep your eyes open over the next couple of months. There, you may be you may want me to come back with some things.
Yeah, I definitely want to, and I can go to see Jim and Lee.
Okay. Cool.
And another final thing is, I think the visualizations are also pretty cool too. Like those circle plots with you know, profiles around it, right? It shows the importance of visualizations too, right?
Yeah, I mean, it really is important, you know, because you know, I'm a believer. I think the visualization strategies are as important as the molecular profiling, all the fancy math we do, and that's what I did my thesis on, was crazy fancy math like that. But you know what? Seeing with your eyes and being able to judge these, and that's the beauty of having spatial data, right? You can actually see stuff with your eyes, and your brain does all sorts of crazy algorithms. And I'm a big fan of that. And one other things we did with the software, which is kind of cool, that not many people actually get initially. We made it so that when you do those volcano plots on pathways and stuff like that, I wanted to be able to click on a biomarker or a pathway in a volcano plot like that, even circle it with a mouse, and it will take you in real time. It takes you to every image that generated that differential term. And that's crazy. It's amazing that we can make it work. So you can do your molecular profiling, you're sitting at your desk, you click on that biomarker, all of a sudden it takes you to the raw image data that's linked to that. And to give you an idea, one of the first times I did that, I clicked on a principal component, which was a really anomalous principal component on a high-plex RNA we were doing. I clicked on that principal component. You know what it took me to? It took me to every tertiary lymphoid structure that was in this patient cohort I was on. And those things are unique immunological features. And no wonder it yanked that particular PCA axis way off to the right compared to everybody else, say. But without that visual thing, I could have stumbled around in that PCA, you know, for a month and not actually realize what the heck I'm looking at. But anyway, that's sort of a bonus thing.
Great, thank you so much. And with this, I think we should thank to Dr. Beechem again. It's clap time. I think I see some clap function now in Zoom.
Hey, it's fun talking with you guys. It's a great series. So I hope you guys keep this thing up. I know it's hard to keep the momentum of these things going. And I know I'm advertising it everywhere I go. All my people know about it, watch it. So I'll keep spreading the word. I love this series, you guys.
Thank you so much.
So much appreciate it.
Okay, see you guys.
And a final thing before we close, of course, we have next week Dr. Wesley Legend from UNC, and we look forward to see you guys. And with this, I think we are closing for today. And yeah, see you all.
Yeah, thank you all, and hope to see you guys next week.
Bye-bye.